Imagine a string with hundreds of beads, each one able to bend and twist in multiple directions. Now imagine this string spontaneously folding itself into a single, precise, intricate shape in less than a second. This is what every protein in your body does, constantly. The scale of this problem is staggering. For an average protein, the number of possible folded shapes is greater than the number of atoms in the known universe. This is the Leventhal paradox. If a protein tried to find its correct shape by randomly sampling every possibility, it would take longer than the age of the universe to succeed. Yet biology does it in milliseconds. Something other than random chance must be at play. For a long time, we could only glimpse these shapes using laborious methods like X-ray crystallography, which involves crystallizing a protein and blasting it with X-rays. But in the 1960s, Christian Anfinsen performed a landmark experiment. He took a protein, ribonuclease, and submerged it in a chemical solution that caused it to completely unfold, losing its biological function. He had destroyed its complex shape. But when he removed the chemical, the protein chain spontaneously refolded back into its original, perfectly functional shape. This proved something profound. All the information needed to specify the final three-dimensional structure is contained entirely within the one-dimensional sequence of its amino acids. The map is written into the chain itself. Anfinsen's discovery created a tantalizing new goal for science. If the sequence contains the instructions, could a computer read them and predict the final shape? To measure progress, the scientific community created a competition called CASP, the Critical Assessment of Protein Structure Prediction. Every 2 years, teams are given amino acid sequences for proteins whose structures have been solved experimentally but not yet published. It's a blind test. For decades, progress was slow. Methods like homology modeling worked well if your protein was very similar to one with a known structure, but they failed on novel folds. The accuracy for the hardest targets barely improved, and the grand challenge remained unsolved. Then, at CASP 13 in 2018, a new kind of competitor emerged. DeepMind, an artificial intelligence company, entered a system called AlphaFold. Instead of trying to simulate the impossibly complex physics of atomic interactions, AlphaFold used a deep neural network. It was trained to predict the distances between every pair of amino acids in the sequence. This created a probability map, a set of spatial constraints that the final structure must satisfy. From this map, it pieced together the 3D shape. It won the competition by a significant margin, signaling that AI was not just another tool, but a fundamentally new approach to the problem. The true revolution came 2 years later with AlphaFold 2. It didn't just improve the old method, it completely reimagined the problem. It incorporated an architecture called a transformer, specifically an attention mechanism, which was originally designed to process human language. Just as a language model determines the relationship between words in a sentence to understand its meaning, AlphaFold 2's core component, the evoformer, pays attention to the relationships between amino acids. It figures out which amino acids, even those far apart in the linear chain, are likely to interact and influence each other in the final folded structure. It was learning the grammar of proteins, building an implicit graph of their physical and evolutionary relationships before it even began to construct the 3D model. The results presented at CASP 14 in 2020 were not just an improvement, they were a shock to the entire field of biology. The gold standard for accuracy is a metric called the Global Distance Test, or GDT, a score out of 100. For decades, a score of 90 or above was considered the finish line, an accuracy equivalent to laborious, multi-year experimental methods. While previous top scores for difficult proteins hovered around 40, AlphaFold 2 achieved a median score of 92.4. On a graph of historical performance, it wasn't a step, it was a vertical line. The 50-year-old grand challenge had effectively been solved. Solving the problem was only the beginning. In collaboration with the European Bioinformatics Institute, DeepMind used AlphaFold 2 to predict the structure of nearly every protein in the human body and released it all for free. Soon after, this database expanded to include over 200 million structures from bacteria, plants, animals, and other organisms. This is like being handed a complete atlas of the machinery of life. Scientists studying a disease caused by a malformed protein no longer have to spend years determining its shape, they can simply look it up. This has already accelerated research into antibiotic resistance, vaccine design for viruses like COVID-19, and understanding the molecular basis of genetic disorders. AlphaFold's success is a landmark for biology, but it may be an even bigger landmark for science itself. It represents a transition in how discovery happens. The traditional scientific method of hypothesis and experimentation is now complemented by AI-driven prediction. The algorithm can explore a solution space far larger than any human or team of humans could, and generate results with astonishing accuracy. This frees up human intellect to focus on the next level of questions, not just asking what a protein's shape is, but why it has that shape, what it does, and how we can design new ones. It's the beginning of a new paradigm, where AI acts not just as an analytical tool, but as a generative engine for scientific insight itself.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.